Okay, so we're going to start this section of the event. We're going to be talking about quality control, quality assurance processes, regulations, that kind of thing. So I wanted to start with introducing the panelists. Dr. Jeff Raber, he's trained as an organic chemist, and he has been operating a facility in California since 2010, measuring components of cannabis and also publishing some of his research results. Dr. Jehan Marcoux, I met him in 2010, and we were both working in the endocannabinoid field. So in addition to our interest in cannabis, we were both doing endocannabinoid research. He got his PhD in the molecular pharmacology, studying the CB1 receptor and binding of different chemicals to that receptor. That was primarily what he studied. His thesis was on cannabinoid receptor in bone as well. And he subsequently won an honor of being the Billy Martin awardee at the International Cannabinoid Research Society meeting. So Billy Martin is the cannabinoid researcher who first defined what we call the tetrad of effects and how THC has been studied in animal models. So Jehan won that award. He now has been working for American Safe Access and as a resident researcher, and also being the auditor for the patient-focused certification program. He's been very involved in developing that program and also working on the cannabis committee at the American Herbal Products Association. And then last but not least, we have Tony Young. He has been an expert in FDA law for about 30 years. And he's been working as legal counsel for the American Herbal Products Association for about 20 years. So if you have any questions about regulatory authority, herbal supplements, dietary supplements, the Dietary Supplement Health and Education Act, he's your go-to man. When the DSHEA came out and there were no provisions for adverse event reporting for these products, and the APA said, well, we think there should be, and the FDA said, well, it's not. We don't have anything to do with that. Tony was the one who shepherded that through and created adverse event reporting for herbal products and supplements. So that's your panel today, and I'm leading it off as the moderator. My name is Dr. Michelle Sexton. You can't really read about me and find out who I am because I'm not in there, so I get to tell you myself. I started as an herbalist, lover of plant medicine. And as a midwife, I went on to go to school at Bastyr University to get my naturopathic medical degree. And during the time I was in Seattle getting that degree, I was working at a laboratory in the University of Washington in the pharmacology department, where we studied the role of the endocannabinoid system in neurodegeneration and neuroinflammation. I ended up doing a postdoctoral fellowship in that lab for 3 years. And as it was coming to a close, I realized what I was really interested in was the whole plant. From my foundations as an herbalist. And at that time, the medical marijuana movement was really growing in Washington State, and no one was measuring components of the cannabis that was being used medically. So I opened the first laboratory facility and have been subsequently involved in creating the standards in Washington State. So that's what I thought I would show you what we adopted in Washington and how that's working with regard to quality control. So what was adopted in Washington State, it's confusing to a lot of people because of just the chain of events in passing legislation. We had passed regulation through the legislature to regulate medical marijuana, and our governor vetoed it. The next year, Initiative 502 passed for adult use, and then came the regulatory authority. And so now medical marijuana is still unregulated, and there's no quality control that's mandated for that product, but that will be changing in this legislative session, and Carrie Borda will be talking more about that. So what I think was a real turning event was early on, right after I-502 was voted in, there was what we called a summit meeting. So it was really the first time that a regulatory body, who was the Washington State Liquor Control Board, brought together a whole panel of laboratories who were doing cannabis analysis, and they came from Colorado, California, Montana, and Washington, and we all sat in a room together. So it was a great experience of the regulators actually wanting to guide public policy based on the science, or what we were actually seeing in practice. And so that formed a lot of foundation of what ended up happening. Okay, now what? There we go. So one of the first things that happened was that Washington State officially adopted the American Herbal Pharmacopoeia monograph standards of identity for guiding the quality control in Washington State. Unfortunately, after adopting it, I don't think anyone ever read it. But we have now gone back, and we recently had a meeting of all the labs, and we've gone back and said, this is what should be guiding what we're doing. Let's go back and see what it says. So I think that's going to happen. And what's also getting ready to happen is the next part of the pharmacopoeia monograph on cannabis is the therapeutic compendium. Because we rushed out the first piece of it for the state of Washington, the therapeutic compendium took much longer. I think it's in its final stages of review, and I'll tell you a little bit more about that tomorrow. But one of the problems that's out there all the time is I can't tell you how many times a week somebody says to me, well, I'm doing an experiment, and I'm taking samples to all the labs, and I'm going to find out the results. I said, well, I can already tell you the answer. So the question is, which one is the right result? So this is a problem, and it's across states, and I hope to demystify that a little bit today. But there's all of these internal things that should happen inside the laboratory. But at the very top of the list, you see sampling plans. The state of Washington would not adopt a formal sampling plan for sampling in the field or in the greenhouse or the grow area. And we were really disappointed about that at the Pharmacopoeia, because it's a foundation of doing analytical work, that none of your analytical work can be valid unless your sampling is valid. So there's sampling in the field, and then you have to also sample it appropriately in the laboratory. So, routine monitoring, basic stuff that goes along with good laboratory practice. And so what the state of Washington adopted was a checklist that's basically for good laboratory practice. And so there's been a lot of confusion in the public, because they think that this certification means proficiency, and it does not establish proficiency standards at all. So you can get certified and still be not proficient at producing results. So the other thing that's really the foundation of labs being able to come up with the same answer is reference standards. So those are our knowns. We have to run those through our equipment. A company has made it, and they've labeled it, this is 100 milligrams of THC, and then we dilute it and make known amounts. But we don't even know that standard that says 100% THC really is, because the bodies that get involved in certifying standards have not yet been involved. So there's all these round-robin tests and things that go on, but in the end, if laboratories are using different reference standards, that's one source of variability across labs. So these are the things that we identified in Washington, and these were topics of a recent meeting that I was telling you about. So this issue of the sampling protocol came up. There's a problem with the definition of potency that I'm going to tell you about. The accuracy in reporting, this reporting to 2 decimal points and the false sense of security it gives you that that's an accurate number. The sample integrity, so what producers are sending as their sample. And again, I already mentioned certification versus proficiency. And then if medical gets swept into this system, there's no screening for pesticides in concentrates, for aflatoxins, for ochratoxin, or anything that could really affect the health of really ill people. So we have a long way to go still. So sampling begins in the field. That's no great secret. In Washington, we haven't gotten yet to the point of, are they going to go out and train people how to sample in the field? Are they going to hire some people to go out and do the sampling? Because the method is everybody picks the crown buds that may represent the highest THC content, and that's how they get it into market, was the highest THC content that they can get. So, you know, we need a standard means of making sure that we have a representative sample of a lot of plant material. A lot in Washington is 5 pounds of plant material. And then, you know, so if you don't sample properly at the outset, you're going to have skewed results and what represents your crop. And then, you know, how do we stop producers from manipulating these samples before they get to the lab? So these are all problems that we're facing. So here was how I tried to make the example to the Liquor Control Board. Because there's so much competition amongst the laboratories, they certified way more laboratories than there is business for them to have. And so, you know, you got to get the business somehow. How do you get your market share? So one strategy is telling the producers, well, I only need 2 grams of a sample. And again, this is back to not adopting a formal sampling procedure, or what is how much does it take to be representative of a 5-pound lot? And we have guidance from European standards and even American standards on sampling batches, how to do it accurately, but none of that was incorporated. We hope to change that. But so if you take only a 2-gram sample for the number of tests that we have to perform in Washington, if you divided it evenly across the 4 tests, 500 milligrams is what would be representing your 5-pound lot. And so then you only have a 1 in 4,563 chance that you might sample that gram that could be infected with a fungus or a microorganism. And we've had about, the last report I got was that 10% across the state were failing microbiology, one of the areas of microbiology that was being tested. I wasn't able to get an update. I've heard reports that some labs say they're having 80% fail rate. In my lab, it's about 20%, do you know? Jeff's got 25%. So that's, I think probably 20% is realistic. This is even with the outdoor crop coming in. You know, and in Washington, even eastern Washington in the fall, it cools off. There's a lot of powdery mildew. There's a lot of fungal growth. So it looks like about a 20% failure rate. But if you go back again to the monograph, the reliability and therefore the ability to comply with state or federal regulations of any conclusions drawn from the analysis of a sample is dependent upon the test sample accurately representing the entire batch. So Roy Upton put this statement in there after the state of Washington failed to adopt a sampling plan, and everybody ignores this. So, you know, as a lab, I instruct my producers how they're supposed to sample a batch and what, you know, we have come to determine would be representative. We still can't get more than 5 grams, though. And I don't think that adequately is representative of a 5-pound batch. So again, if you want to represent the entire batch, how much is going to the lab now? And so here's just a comparison. If you get a 1-gram sample in the lab, a 2-gram, a 3-gram, or a 5-gram, you can see the greater the sample size, the less variability that you're going to have in the result that you give out. So if you're only using 500 milligrams as representative of a batch, reporting to 2 decimal points is unethical, honestly. Here's a paper that was published, and these are just showing again, these are the same plant, the same variety. And so if you sample different plants these were grown under hydroponic conditions in a greenhouse, that you can have variable results. Here's some data from my lab. So here's Blue Dream, just THC, blueberry, Jack Herrera, and it clusters most closely. And I think Jeff has data on that too, that Jeff Herrera sort of stands out from the others often. But there's not a lot of Jack Herrera, I think, in Washington right now. Blue Dream is the #1 selling variety. And then you have the CBD variety. ACDC is really popular. But then you've got the CBD content of that all over the map. And so people may be thinking it's a 20-to-1 CBD to THC ratio, but you never know what you're going to get. So, you know, this is one of the things that we deal with labs, is that just because it's a certain name doesn't mean it has a certain component. And then again, this is just a small study we did showing, crown, middle, and bottom bud, and that's probably a small variability for that when you look at exposure to the light. But you do need to know that Washington has the world's strongest weed, if you didn't know this already. Tongue in cheek, I said that. So this is a quote: "Potency testing, meanwhile, shows Washington's recreational pot is all over the map. It averages 16%, ranges widely. About 2.5 is testing above 28%, and some climb into the 30s and 40s. So when this first started getting reported, I mean, I'm a huge skeptic, and I want somebody to prove that to me. So we're getting ready to do an experiment in our lab to demonstrate that this isn't possible. But also they interviewed John Page for this article, which was great because he's one of the authorities on the genetics, and he said, well, with given the growing conditions, I could say you might see 30 every once in a while, but these numbers for 40s. And I had people call in my lab say, hey, do you guys use that method that gives the higher THC value? And I'm just like, yikes. But these are the strategies the labs are using to get more business. And unfortunately, it's what I call still black market practices, and they need to be cracked down on. So here is an example from a particular lab that sort of got called on the carpet at the meeting for the high numbers. And it turns out that they were getting these high numbers by adding the number for THC acid to THC, and then also adding delta-8 and CBN in there as well. And so apparently this is the method for getting the higher THC content. And now nobody can sell in the recreational market in Washington unless they're way up there. They're having trouble getting it through. So, we're really interested in trying to tame the THC, as Ethan says. And, let's inform the consumer about the whole plant. How do you select your wine? Do you look at the alcohol content? Seriously? So we need to get users to a connoisseur level where this focus on THC is minimized. And so how that's happened, these variations, so NIST is a government organization on national standards, National Institutes of Standards and Technology. They've done things like weights and measures and measuring light and distances and all these kinds of things. And the first time they really stepped in for a biological measurement was when cholesterol started being measured. So how many of you this year have had your cholesterol measured? Raise your hand. So you should, if you're over 40 or 50, you should probably monitor it every year. How many of you feel confident about that number that you get back? Everybody doubts their cholesterol levels. I love it. Well, we should be pretty confident in those levels because there are standards about how to measure that. So there are things you can get blood values for that haven't been through the same processes, especially sort of alternative labs that are out there. But for cholesterol, because NIST became involved, there was 23% variation in what was being reported, and that's been reduced now. So this is what we hope will happen with measuring cannabinoids as we move forward. Here's another paper where they said the potencies were inconceivable, and now this was only an average of about 10 to 12%. So what's getting ready to happen is the Washington State Liquor Control Board they just sent out rules for review where they're defining how do you report this THC number, and it's simply going to be THC, removing the acid mathematically and adding it to the THC acid plus THC, which is how I think everyone was doing it anyway prior to I-502 in Washington State. But even when we went to report these to the state where we had to enter it, it said THC acid, THC, CBD, total cannabinoids. We were all like, what cannabinoids are we counting? And I called the Liquor Control Board and I said, why is CBD acid not on there? And she said, oh, I didn't know there was a CBD acid. So again, they adopted the monograph but failed to read it. So this data is just showing that if you take one sample and you analyze it 5 times, you've got a relative variability of 2.3%. And here's the same, if you take the same lot, this is just 1 pound. And if you sample it 5 times, you've now got even a little bit greater variability. showing the variability across the lot. Oh no, what did I do? Okay. So, my question, and I think I even wrote this in the guidelines to the Liquor Control Board, that potency should only be reported in a range. But it would be great if we could, at the national level, come to agree upon this, because honestly, reporting to 2 decimal points is not honest. If you were running that sample 5 times, and you had several other samples and you ran it 5 times so that you had your average and an error bar, then maybe you could report that average to 2 decimal points. But when you run it 1 time, it's not ethical. So I'm going to talk briefly about contamination. What we passed in Washington came out of the monograph. So if you want to look at the guidelines, they're in the monograph. So many people were passing, and they were just shocked. They couldn't believe that there was any contamination on their cannabis, and they come back, and they're just horrified. And then they show me a picture of the harvest thrown into the back of a dirty pickup truck. So these are people that never grew cannabis before. They were farmers, and that's how they treated other crops. And so they didn't even know that it could be contaminated in the process. But then there were the expert growers who are also horrified that any of their product comes back contaminated. And we do see fungal failures. We see a lot of aerobic and Enterobacteriaceae. And so what we did, because Enterobacteriaceae is such a large group, if you have anything greater than 10 but under 20, we perform this total coliforms, and if you have under 10, it will still pass this, and all of it has to have these, and there's 0 tolerance right now, and we're looking at relaxing the limits on that a bit. So here's a story that just came out. They recalled some of their product. It was contaminated with Enterobacter, not associated with acute health risk. Well, which Enterobacter? It matters. And we don't really know the health hazards. There's reports of contaminated marijuana being inhaled, and people with compromised immune systems, that's more with fungus. But, we really don't know a lot about this contamination and how this product's being used. But what we did say in the monograph was these values do not specifically represent pass or fail criteria, but that's exactly how the Liquor Control Board interpreted it, and they put these numbers in there as pass or fail criteria. And now I don't know if we'll be able to get them to revise that. Right now, this is not the most important thing on their docket. Rather, they recommend levels when plants are produced under normal circumstances. Individual herbs such as mints, which have a high concentration of trichomes, such as cannabis, so you're going to have higher levels of molds in seasons of heavy rain. Washington is constant rain. So, I think we need to look at relaxing some of these limits, perhaps. And then what about the sample integrity they bring in for microbiological? So what some labs have been reporting is that people are bringing in separate samples and saying, oh, here's my potency sample, and then this one is my microbiology sample. Or they're submitting it already ground up, so you can't really do a good visual inspection of it, which is part of what it has to pass visual inspection. And then what the monograph says is that none of these organisms are found in 1 gram. And people are using other methods of detection where they're looking at the DNA, and as far as I know, those platforms haven't been validated by any third party for a certain amount of material yet. And so again, if they're only sampling 500 milligrams, you saw what the chances were of it being representative. The AOAC guidelines say, so if you're going to validate it, you should show less than 5% false negatives and less than 10% false positives. So some of the methods that are out there may haven't been through very stringent validation processes. So what are these processes? We've been talking about this in-product testing and the output here. And we need a lot of things to get to a point where consumers can trust this. In the meantime, realize Yeah, there's a lot of variability. And if you want to waste your money and go take it to 5 labs, do that and just count on getting 5 answers. And it's not fair to pick the one that's the highest because you don't know what the right answer is. And we don't even know what the right answer is. But also this depends on everything that's happening on the back end of the input and the crop. And Jay Hunt will talk a little bit more about that. So how do we address this variation? We need finished products that have predetermined quality and reliability. We need that for this industry. We need methods validation. The organizations that typically do this didn't want to have anything to do with it because it's a Schedule I drug. We hope those things are changing. There's international committees for consensus process. We need proficiency testing, but we can't really do this until we have certified standards and validated methodologies. So don't count on knowing whether your lab is proficient or not anytime in the very near future. And we really need a team approach, and this is what we're starting to see happen, that professional analytical chemists are coming up and taking an interest. Everybody's willing to come out of the closet now and say they're interested, and this includes the American Chemical Society, the American Oil Chemists Society, and many others. But we need government agencies and professional organizations to continue to come together in unity to really create everything we need as a foundation for quality assurance and quality control of cannabis products. So thanks for your attention, and at this point, I guess Jeff is next.
Thank you, Michelle. I'll just quickly echo some of her comments. We also run a laboratory in Washington. We started in California in 2010 and opened up in Washington a little over a year ago. And we got a sample from a client and said, don't hand this back to me with the result unless it's over 20%. I said, do you guys know how the labs work up here? It was very alarming to me, but I think I certainly can comment and say my frustration is the same as Michelle's up there. It's exceptionally difficult to operate with a number of other labs who are just trying to find the absolute highest number, not trying to operate properly or ethically, and insist on running in the race to the bottom to try and do it as cheap as possible, actually below some costs, is what I think is actually going on. So it's very unfortunate because it would be better for a patient to have no number than the wrong number, in my opinion. So it can be exceptionally misleading and can actually be detrimental if someone thinks a potency test represents a quality control test for pesticides or bacteria and none of that was done, or if they miscalculate a dose or believe there was CBD in there when there actually wasn't. Those things can be exceptionally harmful to a patient. So I think it's the most responsible position in the game is to be a testing laboratory. And it takes great integrity and a lot of technical competency to do it very, very well. And you're challenged everywhere and every angle possible, from clients to regulators to lawmakers to producers to patients. Everybody is really looking for that number. And if I had a nickel for every expert that was the best grower out there, I swear it would be, we'd have a really fancy lab. That would definitely be the case. But it is really an eye-opening position for many, and it's very difficult. It's the only job that you can actually do very well at and end up losing all your clients. And so it was a shock to me. I was always told in school, do well, get A's, study hard, and then if you do your job right, you'll actually be able to continue doing that. Well, if you do the job right in Washington and you give a potency number, a THC value that's accurate, it was sampled properly and representative, and you say here's 16%, you'll never see that guy again. So it's very disturbing. They did not listen to us when we first sat down and said sample integrity is probably the most important position. And if you don't have an independent representative taking that sample, you're going to have issues. And when they sat us down a year later, they said, we don't know why we have all these issues. We said, well, we told you so is really the only answer that we could give you, but here's how you can correct it, and please listen to us this time. We have worked with some of our clients up there to actually develop sampling plans, and we make them do a number of samples so that you can see that there is integrity in how they're doing that. But I think it would be much better to certify results if the labs or some other independent agent was actually taking the sample. You can have all your equipment calibrated, you can run it the right way with all the right people, but if you don't have the right sample, you're getting the wrong number in the end anyway. So it has to be representative of the entire picture. So that being said about testing labs, I thought, since I'm kind of bored myself personally talking about some of that stuff, but I know it's very important, that since we're here talking about regulations, laws, and how those can be shaped, that I would give a little bit of a perspective on some of the data that we've collected on other studies, and hopefully using that towards shaping regulations. I've spoken a lot inside of a lot of regulatory bodies from Connecticut, Nevada, Illinois, Colorado, and Washington to try and help them craft regulations and understand how testing can be done, what you can test for, where it should be implemented in their program, and which types of agents they should be looking for and how. And I think only using insight and valuable perspective that we've gained from operating in California for a large number of years, because since there are no rules, you see absolutely everything and anything. So we've got a very broad-based perspective on all the types of different products someone might try and put cannabis in, and just picture any food. We've probably seen cannabis inside of that. And we also have a Dutch national citizen. He actually married an American woman and came to the US, and he helped set up the Dutch National Medical Program. So we are very fortunate in our lab to have Sietse Elzinga, an amazing scientist who actually really understands cannabis very, very well, brought 8 years of Dutch cannabis experience into our lab in 2011, and we certainly have benefited a tremendous amount from his intellect, his understanding, and his knowledge base coming from the Netherlands. So setting those standards and protocols has been, important in helping regulators and other types of lawmakers understand what they need to know about labs, and something that's very technical has certainly been an interesting challenge for me. Most of them were just scared to death that cannabis is even coming across their desk to begin with. And then they hear this thing called labs, and they have no idea what a lab does. And then the words start to get longer, and the multiple syllables show up, and they're like, oh man, you can see the eyes glaze over, and they're really lost. So we have to slow it down, dial it back a little bit, and actually give them some copy-paste language. And I think we're getting better to a place now where there are a number of examples and models, and I think that's been very helpful in seeing these things develop. But the first ones down the path they had a very, very difficult job, I would say. So let's see if we can. So highlighting some of the picture across the country, you'll see that there are a number of states that require mandatory testing, and I think that is definitely the trend. It's here to stay. It was funny when we started in 2010, some of the comments we got were, you can test this. Like, it's just a plant. Yep, it's not some foreign mystery material from the moon or something like that. Yep, it's very easily tested. And you can, in fact, test it in a large number of ways. What you want to test for, how you test for it, how fast and accurate you are, and what you're representing in that sample and that test that you're taking are the important parts to consider. So I think now we see a couple different states doing things in different fashions. We're seeing that federal experiments within the state laboratories and getting a chance to, oversee a couple different models. It's certainly a very interesting case from an experimentalist like myself. And if you look at places like Colorado, I mean, they were one of the early pioneers. Massachusetts has an incredible sampling plan that they've included in their regulations. And Connecticut set the grade for the definition of pharmaceutical-grade medical cannabis. So their specifications are plus or minus 3% of the label claim. And that is extremely stringent for any botanical product. So it actually ends up requiring homogenization and very diligent sampling to make sure that you can actually get that number that will be placed on the label, so that if they go and take the sample off of the shelf, that they should end up with something within plus or minus 3% of that value. Those states require cannabinoid potency testing and also full safety screens, including heavy metals and pesticides, microbiological contaminants as well. As Michelle was saying about Washington, they kind of said, well, we'll just get started. We'll point to this monograph that we won't read and just hope someone follows that, and we'll go down that path for a while. They did listen to the fact that it is an inhalation product, and that is much more sensitive to the body than orally consuming. You don't have first-pass metabolism. You don't have stomach acid breaking things down. You don't have different absorption rates. Inhalation is just like injecting it into your bloodstream. So it's a very different health and safety concern than it is for any food product that you would orally consume. So there, the major requirements are foreign matter inspection with a visual inspection, moisture analysis, which is just a kind of quality control check, microbiological contamination, and cannabinoid profile. And they didn't put CBD acid in there because they didn't think standards were available for that. So it's fortunately they are now. More of these reference standard companies are producing more and more cannabinoid standards for this purpose, and we're starting to see that evolve. Certainly 5 years ago they were scared to death when I called and asked, can I get some of these things? And they said maybe for $10,000 for a couple vials we could send that to you. It's come a very long way from that. Oregon took a very interesting approach where they said in their medical system so far, dispensaries are required to test for pesticides, mold, and mildew. They didn't know which ones, they didn't know how, they just said that was the only words that they could come up with. We've seen things similar to that in terms of Los Angeles and Long Beach at one time had considered city ordinances to that effect, but they were repealed after 200 lawsuits rained down on the city of Los Angeles. There was no way that one was going to stand that battle. But they still aren't quite sure, how to define the word pesticide. What exactly does that mean? It can mean thousands of chemicals to a chemist like myself. So I certainly cannot go and tool up my lab to test for that many in any sort of cost-effective manner. And the equipment to do so costs more than a house for some people. It's upwards of $300,000 to $500,000 depending on the approach you actually want to take. So when someone says, why can't my pesticide test be $50 and why do I have to do it all the time? They're usually $500, and they're done much, much more frequently, so that the cost of maintaining that equipment, which can be upwards of $30,000 to $50,000 a year, let alone the people to run it, can actually be paid for at an economy of scale. So getting those things started or sitting there with that equipment and watching no one use it is going to kill a lot of startup businesses and can be an exceptional challenge. In California, as I mentioned, it's all voluntary. So what we've been able to understand and learn there has been through a lot of hard work and a labor of love. We drove 50,000 miles a year for many years to try and go and collect samples by hand so we could understand exactly what was going on, and that's how we generated a lot of useful data that we use. We've since trained a number of people how to do it, and you have much less of a homogeneity concern when you have a concentrate sample. That batch is usually pretty homogeneous. It's not like the flowers, so it's a little easier to do things of that type. As we discussed, the amount of microbiological contaminations that we see, we've seen in that scenario, we've taken the samples, we understand what's going on, approximately 25% of them fail our gold level, and that's the same level that's set in Washington's I-502 recreational market. And it's mostly molds, but sometimes we see the entire bacterial party. It'll fail every one of the screens, total aerobic count, enterobacteria and coliforms, and the molds section as well. And there are some published reports out of the UK, I believe, that talked about aspergillosis, which they attributed to contaminated cannabis being inhaled. So I believe that can be a concern. Pesticides, and I think in that word pesticides, we try and look for pesticides, herbicides, fungicides, and plant growth regulators. Plant growth regulators are exceptionally prevalent. They're used to manipulate the plant and maximize the return in the shortest amount of time and increase the yields. Those are definitely prevalent, and sometimes unbeknownst to the cultivator who thought that they were just using nutrients, which in fact had those types of agents in there. 10% of our tests are found to fail for that. And I need to caution everybody, when you hear a lab quote the numbers or percentage of numbers that they see, we're given the samples that people thought didn't have any pesticides on it. We're not seeing the entire market's worth, so it's not a very broad-based picture. It's what's come into the lab. And we have had numbers of dispensaries say when they tell potential producing members of their dispensary that they're going to have their products tested with our laboratory, they pick the bag up and run back out the door. They know that we'll catch it. We're really good at what we do, and therefore they're just going to go down the street to the next guy who doesn't test in California. So we're certainly not seeing the general case. We actually went out private, secret shopper style, and circumvented, some of the interest in that result. And we looked at a number of dispensaries in a small area in Southern California and found 35% of them contain pesticides or plant growth regulators. So it can be a very large number. And you do concentrate pesticides into the concentrates. So I think that's not generally well understood. If it's on the plant to begin with, most likely you're going to concentrate it, and it will actually result more clearly in the concentrate. It's a little easier to find in the concentrate. The matrix is a little simplified. But it will be there. There's I don't know of any methods possible that would actually destroy or degrade any herbicides, fungicides, or pesticides like that. A lot of them have very similar properties to THC and the cannabinoids you're going after, so they're just going to go along for the ride and end up being concentrated as well. So producing medicine in that sense, you haven't clarified all of that. You can get rid of microbiological contamination with things like CO2 or large amounts of ethanol. And that will actually, kill any live bacteria, but it may leave behind aflatoxins and things of that type. But pesticides are a whole other story. You're just better off never having them in the plant to begin with. Residual solvents also are often found inside of concentrates from butane, hexane, isopropanol, acetone, ethanol. We've even found benzene and traces of other petroleum cuts that were used, such as naphtha and things of that type. So just because it's a concentrate doesn't mean it's clean or any better. And I think it is a word of caution that even though we can do a pretty good job at what we look at, we are not able to look at all things possible. So in California, when they say, good luck, we're not even going to tell you what to look for. That's one challenge. The number of residual solvents you'd have to look for are large. As I said, pesticides can be thousands of molecules. You don't know what to look for there. You don't know what's allowed to be applied, and you don't know what they may be using. We've used our best judgment in terms of what might a cultivator get on websites like Rosemania or go to Home Depot and just see what's readily available that they could get from a hydroponic store or those types of ornamental places, which you shouldn't use for consumption, but we know they do. And just are trying to play the cat and mouse game. We try and not publish our pesticide list so that people can't circumvent that and use something that we're not looking for. So as states come around and say, you're allowed to use these, labs will have to test for those to say they're at acceptable limits, but they'll also have to test for those things that still may be used that are outside the system. It's a lot like athletics and blood doping and things of that type. It's a very, very challenging and complicated game. So it's certainly, as I was saying, a very, very complex problem. There are a number of things that you need to look at and pay attention to, and a number of complexities that go along with that. And as a lab in a voluntary market, there's only so much equipment that you can afford. And as you do better and better, you get less and less clients. So it's a delicate balancing act to try and say, I can afford this one, and I'm pretty sure people are going to be protected in some respects better than nothing. But I still would like to have much, much better hardware and be able to do a bigger, better job. Hopefully someday that'll come. Certainly with mandatory testing, we believe that would actually be the case. In the state of Washington, they still aren't sure which pesticides they're going to monitor or how frequently they're going to do that testing. So we have simply not bought any equipment for that in terms of waiting. You don't want to make a $100,000 decision when you should have made a $300,000 one. So it's very challenging from that respects too. So it's method development takes a long time too. To do that right, to do it repeatedly, and to do it really, really well, it takes a considerable amount of efforts. So it would be another word of caution if a new state comes online. It'll be interesting to watch in Nevada how fast a lab says that they're ready to go, especially if they were a brand new lab that never did any cannabis testing before. So if they're ready to go day 2 that they've opened their doors, I'm really going to question that one, because it takes a lot of practice and experience to say that they've done that well and really went through the whole picture. But we know the market doesn't want to wait. So what I'd like to talk about now is our pesticide inhalation study that Dr. Rousseau had alluded to earlier. And I think one question we got when we started testing pesticides was, how do you know that that would actually be a problem for me? How do I understand that if I have it on my plant material that I would ingest that? And that's a very fair question. I was like, that's one of the best questions I've heard operating in the space so far. I said, that's good. We got an intern from the local community college and sicked him on the project. A very bright young man, Nick Sullivan, and said, here you go. We're going to set you up. And we studied the pipe on the left, the water pipe that was produced by Scientific Inhalations. They supported the study. So they said, how about you study our device along with a typical handheld one on the right, which they made specially for us to configure to connect into our equipment. So the one on the left used either no filters like you see pictured, or virgin coconut carbon in the top and cotton on the right-hand side. So those were the 3 types of studies that we did in that. And this was the basic qualitative study that showed there's a number of different pesticides that you could put on there and screen, and that you can see that there are varying amounts of those that were available. The green line is the one with the filters, and that actually was helping to alleviate exposure to patients. So it would probably be a good idea to use that. And it's the same reason you see cigarette filters. So in the cigarette world, they understand that pesticides may be present, but they really don't care because they think they're going to kill you anyway. And that was, that was the rationale. So there are no pesticide standards in the tobacco world. We can't actually even apply that here. But we're talking medicine to people who are immunocompromised. They may be very sick. They may be very young and developing. And we're not trying to cause harm to anybody. We're actually trying to help people. So it's a very different, story and a very different case. But you can see even with that approach, you can't stop everything from coming into the airway. So we focused that study to do the quantitative study, which is a lot more difficult and a lot more rigorous to do inside the lab and takes a lot more time. So instead of doing that broad-based number of components, we chose four of the more commonly used ones that we've seen inside of the lab when we've done the detection. And those four are pictured here. So things like diazinon, bifenthrin, paclobutrazol, the plant growth regulator, and permethrin. And what we found was in that glass handheld pipe, which would probably be very similar to a pre-rolled cigarette or any other typical water pipe, you can have up to 70% of that molecule entering the airway and hitting your lungs. Now, it doesn't mean you're going to absorb all of that, but you're definitely going to have a good chance to absorb all of that. That was an alarming number. I mean, I think most of us had first assumed that there's going to be some sort of degradation, certainly when you're using combustion, because it was placed on the plant material, and then we hit it with the lighter. So you can put it all on the plant material, make sure you take off all the solvent, quantify that it's still on the plant material, put a known amount of plant material inside of the device, and then use a lighter, and we've connected it, and I have a diagram of it later, to a mechanical lung system. So vacuum pumps, and we had trained ourselves at an expo. We had a whole bunch of people just come up and inhale water up through a column so we could see how strong inhalation rates actually were. So we used a little bit of a rational approach, not just something that's going to pull really hard, but this is based on a number of users saying that this is most likely what the inhalation volume is going to be like when someone at home goes and uses this. And what the results showed were definitely quite alarming. So even a typical water pipe, that's the one in the middle, really will expose you to that. You don't see much condensation in terms of water or anything else cooling. Only along that cotton path is what we found actually condensed a large number of pesticides. There's a lot of surface area there, and it actually holds on to them pretty well. So it makes rational sense. And we did publish this. This was our first peer-reviewed publication in the Journal of Toxicology. So, and it was really nice for an intern to earn a publication out of his job, too. So fortunately, we hired that guy, so that was really good for us. Microbiologically, we use a rapid microbiological testing platform, and we can look at total aerobics, enterobacteria with coliforms or combined yeast and molds. This platform looks for any live organisms, and it can do mold testing in 48 hours of test time. So it's rapid enough that people could wait a little bit. If you have days and days and days of running your test, are you also growing mold and bacteria inside of the samples while they still sit there? So it's a little bit of a challenge. And our levels, we call gold. We try to make it a little bit like a competition. They always say they're the best, so we thought if we gave them gold stars or gold, silver, and bronze, it might actually be helpful to them to try and compete. And the gold is based on USP levels for dried botanicals. So we thought that was reasonable, and fortunately, that was exactly where Washington landed in the recreational market. It's still debatable as to where a rational level should be for medical purposes. We're not going to be irradiating anything here. I don't think anyone's going to do that in the United States. No one's getting a license for that, and I wouldn't want to see these guys all getting that either. But to really understand, what level makes sense, we're going to take the same methodologies that we've used for the pesticide part and are now performing that study with live bacteria. We've got a collaborator that's actually put fluorescent tags inside of the bacteria. It's kind of cool, so you can actually see them glow and real easily. And then we're going to quantify them as well and see how much can we place on plant material and how much might be going into the lungs of a potential user using these types of methodologies. So this is a description of the system. And this was a study that we used on pre-rolled cigarettes. We thought, a lot of states, at least 2 of them now, Minnesota and New York, have said we don't want flowers around, because they apparently don't know what a dab is. So I think they were thinking that there was no recreational problems if there's no flowers, but I think that they're just misinformed. I certainly understand the utility in concentrates being medicinal, and maybe that was their intent. But I don't think that you should throw flowers away because bulk plant material can be exceptionally useful, used in tea for the cannabinoid acids. It can be very helpful to a number of people, and some people still prefer that, or that's what they knew before. So I'm really kind of libertarian-minded. It should be an open choice for anybody and everybody. We shouldn't have CBD only or THC only. You should have whatever cannabinoid works for everybody. Every form of the plant should be available, and you should let all the patients make that decision with their doctor, not a lawmaker or a regulator should not be making that decision. So I'm going to try to use science to convince them of that. So wish me luck. It's definitely, we thought some rational-based approaches. We had seen some cases, and it's always a concern how much actually might get into a consumer if they do have a standardized pre-rolled cigarette. So we thought, hey, this is another good project for someone. And since we have a system, let's go ahead and do it. And let's try and do it to the best of our abilities and capture everything possible. So we have a vacuum pump with a time controller, and that can actually, puff and do the inhalations. And you'll see the gas traps there in the blue, and there are two sets of them. One was to get the mainstream smoke that comes out the end of the product, the end of the pre-roll, and then the other one was to capture all of the side stream stuff. So what you understand in early pilot studies is, man, I can't find all the THC or CBD. Where is it going? So it's not in my traps, and I've had enough of those traps to know that I wasn't missing anything. It was all being condensed and trapped. So it's not there. It's not inside of the remaining parts of the ash. There's only very little bit there. Where else might it go? So that was why we decided to capture some of the side stream smoke as well. And we do find some of it there. But ultimately, some of it just gets destroyed. So it's maybe not the most efficient means of converting THC acid or CBD acid into THC and CBD. And that was, I think what we ultimately came to. This was also published in a peer-reviewed article, the Natural Product Chemistry and Research. It was just recently published earlier this year. And they said this was the most comprehensive study of this type that they had actually seen, and we did not include any tobacco in there as well. So others had done some with both rolled together, but that's a very different temperature, and that doesn't necessarily represent exactly what some sort of patient might use at home. And what we found is a quick summary shown here. So on the left, you'll see the average across all of them in the mainstream smoke, and it's about 35 to 40% of what's available in that pre-roll could actually be inhaled by the consumer. So even though it's not a lot, it is consistent. So we did a number of studies. We did 3 different plant materials. One was about 14% THC, one was 15, and one was a little over 19. And we also did it with CBD mixtures, and you could tell by a linear prediction that you knew exactly how much you would get. So it was reproducible, which means you could use that in a medicinal fashion. And it was certainly quite interesting to see that we could reproducibly generate that result. Now that was, of course, a machine doing that the same every single time. So if your patient individual at home could be trained to do that the same every time, they could get consistent results that way. As long as you standardize the medicine and the product that you give to them, and then they standardize their use, I think you have a great ability to say it's a standardized medicinal product that they could use as needed, understanding that they're going to get a certain number of milligrams per inhalation once you know how many inhalation units they take per pre-rolled amount. And I think that's pretty much the basic summary. I don't want to take everyone else's time, but in general, we've seen a lot of different variations in terms of how you should approach regulators. I would say from my perspective, we're hoping to use science and understanding to say how you can use rational information and really help everyone understand that pesticides are a problem. I believe microbiologically we're going to see much the same thing because early reports are very similar. And really don't throw the botanical product out and have only one form or another. We should really allow patients and doctors to make the choice. And I'll turn it over to you.
Okay. Okay. All right. We've heard a lot today. We heard from 2 people who operate laboratories that are certified by patient-focused certification. But what is quality assurance? What is this method development stuff? Why do we need these standards? At first, I was going to give a talk that was very focused on things I had seen while being the chief auditor of the patient-focused certification program, but I realized there's an awareness issue. There's a little bit of an alphabet soup problem with terminology. I'll give you a quick example. Was at the American Chemical Society meeting last week in Denver. They invited an extract manufacturer to address many of the scientists in the Chemical Health and Safety Division. When asked if he was following any if he was GMP compliant, the operator's response was, I think so, which is probably the worst response you can give, which would be better to say nothing at all at that point. So I think he didn't even know what GMP was, so there's a big awareness and outreach issue. So what I'd like to do is just sort of lift the lid off of what constitutes general requirements for good laboratory, good manufacturing processes. And so basically I'm going to talk to you a little bit about a paradigm shift that Michelle Sexton talked about towards the end of her talk. I'm going to talk about quality control and what is quality assurance, quality control, method validation, as well as discussing good manufacturing processes. So here are my axioms for auditing or quality control for the things I do. The first one is no assumptions. Don't assume anything. If you're taking in a product that you're testing for pesticide, treat it like it has pesticides on it. When you're developing your methods, go from simple to complex. Don't immediately try to shove a brownie into your liquid chromatograph. Start with THC, see what possible matrix interference there is. Go from simple to complex. And the third and probably most important one is use brackets whenever possible to give ranges and actual real values. Those are my three axioms, and I'll be periodically referring to them. So what constitutes best practices? Really, quite simply, it's made up of three things. You have science, technology, quality assurance, and quality control. Science moves very, very slow. Technology moves extremely fast. But they're both balanced out by quality assurance and quality control. And so these are sort of just the baseline of how you start thinking about best practices. And so we've experienced a paradigm shift, not just in the cannabis industry, but in the FDA and quality control in general. We go from reactive to proactive. The old process that still is around today is variable input, fixed process, many scrap batches. So what does this mean? It means you have variable input. You're getting in a raw product, you're blending it, you're mixing it, you're preparing something, and you have a fixed process, which means you have a QC, QA test at the end. So you've invested who knows how much money, time, effort into generating a product, and you QC it, QA it at the end. It doesn't pass your standards, you have to scrap it. Extremely inefficient and wasteful. The new paradigm is variable input, variable processes, which, if you're making something, before you go from step A to step B, maybe you should make sure step A was done correctly. And then when you go to step B and you're getting ready to go to step C, make sure step C was done correctly. Don't wait until you've already made the product to go back and QC it. Variable process allows you to create more consistent output. So quality control. Basically, this ensures that data information generated are of a known accuracy, degree of probability, and proper documentation and training. Quality assurance does incorporate quality control procedures, but there's more accountability for the results. Someone has to sign a piece of paper saying these are the official results from this test. There's traceability of an analytical report. Who was operating the machine that day? Who was doing sample prep? Proper and calibration of standardized procedures, method validation, all this stuff comes down to documentation. So are things written down? And so when you have a QC, QA system, it basically represents a pattern of actions that's for optimization, productivity, communication, and generally increasing value. All right. So general requirements for laboratory quality control. So trained personnel. This is a moving target. So you want to make sure your personnel are trained. But if you upgrade your software from, from whatever was previous to ChemStation, are they trained in how to use that software? If you get a new piece of equipment, a new HPLC machine, a new version of it, you have to also make sure that your method is robust enough that it can handle changes in equipment, but also people are trained on that equipment. Environment and facilities are properly controlled. The environment's controlled. It's safe. It has things like a fire exit. Solvent storage is probably the biggest issue that I've encountered, and I can't stress this enough. Reference materials, getting appropriate reference materials for what you're studying. Having sampling guidelines, sample handling, sample storage, calibration documents that involve with calibration. Also, how you calibrate it, measurements of uncertainty, precision and accuracy. So, you know, how close are you getting to the target, and how many times how reproducible is it? As well as one of the important parts of method validation, and you could ask this of any laboratory, and they should be able to give you an answer, is what is your limit of detection and what is your limit of quantification? If they haven't done this basic stuff to calibrate their equipment, I wouldn't necessarily trust the data. So you want to know how low they can go and still detect, say, THC, and you also want to know how low they can go and still get an accurate quantification of an active ingredient. And also, you have to think about qualifications for using methods and the people who work there. Here's an example of a fail. So this is on the manufacturing floor of a non-PFC certified manufacturer/laboratory. First, I'll draw your attention to the exposed septic pipe on the manufacturing floor here. But this is all a flammable solvent stacked up into the wall in huge jugs next to an oven. So this pretty much ends with some guy running out of the building with his dreadlocks on fire. So there's actually, the FDA actually goes around and inspects places for this. And I know that many of us are critical of the FDA, but I thought it was important to point out because this is all public information. You can go look up 483 non-compliance issues. And people do get cited for no written procedures for their HPLC system or validation on hardware or software. No audit trail archive. You know, issues with data acquisition and storage. You know, not validated. No exchange between hardware and software compliance. So this is a really important component. And this starts as, you know, much like a PFC program, a simple 1 or 2 compliance issues isn't a big deal, but if you see a lot of them, it represents a systematic error with the management that doesn't know what's going on in their own facility. So it's really, to think about it, this is a lot of standards are around method validation, making sure you can prove what you're doing. So these are just some terms about method validation you should think about. Specificity, how specific is the test? Linearity, and this is in range. This is something our previous speakers has touched upon. So you have this calibrated piece of equipment, and you want to know how many points they're looking at. What is their range in that calibration curve? And I can give you an example of why this is important. And this gets back to something called Beer's Law. And you could have a whole panel at this conference about what is Beer's Law. But the quick and simple version is if you have 2 points anywhere, it's very easy to draw a straight line between them. But the more points you have, the less straight that line becomes. In the conceptual way and this is important when you're generating calibration curves or you're doing reproducibility studies. And a simple way to remember Beer's Law is if you go to the bar and you have 2 drinks, you can still walk a straight line. If you have 12 drinks, Beer's Law. So accuracy and precision studies are important to conduct. So not just whether or not you're getting the right answer, but how close you're getting each time you do it. We discussed limit of detection, limit of quantification. Robustness is a very often overlooked thing, especially for expanding laboratories that want to have operations in multiple states. How well do the methods transfer? Different operators, different equipment, slightly different changes in solvent can all affect how well a method transfers. I've run into this problem in biochemistry where you ship someone's cells and the cells don't grow in their medium. But this could also work if you're trying to, say, transfer an HPLC, a liquid chromatography or potency analysis, to another state or another country. So paying attention to those variables and knowing in advance where the weaknesses are in your system. Also looking at system suitability. How good is your separation, resolution? Do those little peaks look nice? Are all the peaks able to be identified? And so this is an overview of the validation process. We're not talking about A to B to C. We're talking about a circular process that feeds into each other. And when you're starting to validate your method, we want to think about starting with questions. What's available in the literature? What do you already know? Pick your instruments. Look at your response to your analyte. Minimize overlap with other compounds. Start to look at matrix effects. And once you've begin with questions, designed your sort of experimental setup. You've tried to optimize it a little bit. You go into sample prep. The simplest version of sample prep is dilute and shoot. Put your product into a liquid, dilute it, take some out, put it in the machine. This doesn't always work. A lot of edibles tend to become really gooey and sticky, so there might be some sample prep methods. But the idea is you can go back, optimize, choose different equipment, ask better questions. But by the time you get to a validated method, you better have a linear response, Beer's law, law of proportionality. You also must be able to identify any impurities that come up. So are all the little peaks in a chromatogram identifiable? Do they know what they are? Or they're just like, I don't know, we'll just, like that's mystery compound A. But that should not happen. And this is a circular process. I want to just think about it. It feeds into each other. So what about good manufacturing practices? Well, GLP, GMP, there's some overlap there. Basically, very similar stuff. You want to make sure the premises and the buildings are appropriate for what you're doing there. You're zoned appropriately. If you're operating in a residential area and you're only allowed to have one employee, you should only have one employee. Personal protective equipment. It doesn't do any good just to wear shoe covers if you're not covering your clothing or your hair. Personnel and materials that are allowed into the place need to be checked and verified. Production and process control, and having your QC/QA programs, qualification and validation of methods, documentation and labeling. Holding and distribution is also a really important thing. Do you have transportation guidelines for how your materials are supposed to travel when they're stored for prolonged periods of time? And now I'm going to show you an example from Vice magazine of a cannabis ice cream manufacturer. And I can't tell you how many safety code violations in this picture. So before we get to the centerpiece, I just want to point out that the camera crew is not wearing any personal protective gear. This is a baseball cap, not a hairnet. He's not wearing shoe covers. Their dreadlocks or whatever are exposed, or long hair or whatever. I used to have a ponytail, but I wore a hairnet. And this is not what we mean by sampling procedures. Also, the top of the ice cream machine is open. This is what when we talk about PFC certification, I think this is a good difference of what you would never see in a certified place. So basic product safety handling guidelines, they're pretty important to follow. So how do you initiate these sort of practices? This little flow chart. So let's say you're doing what you do. Well, you should be able to prove it, right? And if you're unable to prove it, you should ask why. And you should also say what you do, document it. And all of these things feed into continuous improvement and innovation in what you're doing. And so if you're unable to prove something, or unable to do what you do, or unable to describe what you're doing, corrective actions, preventative actions, document trails, method validation, these sorts of things. This all leads to innovation. So I'm going to give you a couple examples, a little bit of Debbie Downer time, and just a heads up, I'm glad you all are done with lunch. So we're going to talk about some quality control methods first in general, or fails. I'm going to discuss some food stuff that has failed recently too. So I thought this was appropriate because glycol is a very popular component of many cannabis products on the market, but glycol can become contaminated. And there's been 3 instances I was able to find where contaminated glycol with diethylene glycol has led to the deaths of, I think, almost 300 children since 1969, when various medicines were prepared and there wasn't good quality control procedures in place checking the quality of the product at each step. We can point to many, many, many different quality control failures throughout history. Challenger was due to a gasket that hadn't been properly QC'd. Aspirin cyanide poisoning, which killed a groom, his brother, and their daughter on their wedding day. Vioxx, thalidomide, which wasn't properly tested in pregnancy studies before being released to the market. Ford Pinto, the iPhone 4, the Hubble telescope. And if you're savvy enough, you might even be able to name a cannabinoid that wasn't probably properly QC/QA'd before being released on the market. Rimonabant, thank you. So that was the anti-CB, it blocked the CB1 receptor and was pulled for side effects. It wasn't properly studied or studied enough. So some other QC failures. I like this one a lot. Green, right? That's a fail. So just going to finish a couple images to drive home what I mean by quality control. That's a frog. You know, unevenly filled liquid containers in a supermarket. I think this could be important when you're thinking about vape pens or tinctures or things like that. And it's very rarely that quality control mishaps work out in your favor. So here is hamburger buns and hot dog packaging. I guess it doesn't matter so much sativa indica, right, Ethan? But if there's immersing content different, watch out. So this is the one instance I could find where quality control mess-ups might fall under your favor, is the giant Oreo. But these are all examples of things that can go wrong, even with good quality control procedures in place. And this stuff usually happens when there's only like 90% conformity. So, you know, the PFC program is centered on 100% conformance to the guideline. And this is important because for other industries, if you only allowed, say, 99.9% conformance to their standards, it would equal, say, 2 unsafe landings at Chicago Airport every week, which would pretty much shut down the airport. 16,000 pieces of mail to be lost each hour by the post office if they only followed 99.9% of their SOPs. Or in terms of pharmacists, 20,000 prescriptions to be incorrectly filled out each year. Less fun than it sounds. So pick a standard, pick any standard. There's lots of stuff out there. There's FDA, ICH, PFC, USP, ISO, AOAC, AOCS. There's lots and lots of organizations that have proposed standards for proficiency testing, and many of them overlap. For instance, the material used for PFC, like the American Herbal Pharmacopoeia Cannabis Monograph, is USP compliant. ICH overlaps with FDA and USPA guidelines. And make sure that whatever you're doing for your business or your operation, that it's appropriate for what you're doing. ISO is one of my favorite things to see floating around the industry. For instance, if a laboratory was ISO 9000, would that be appropriate? Not really, because hotels can be ISO 9000. You know, you want to make sure it's the proper alphabet soup lettering that you know, ISO 17025 is specific to laboratory processes. So you want to make sure it's appropriate. And standards are not there to hide things. It's to bring things to light and to make things better. These are not, you know, illusions or tricks to get past regulatory agencies. It's about transparency. I'm going to just go into a little bit about batches. This is a shameless plug for Monday, where Kristen Nevedal, who's the PFC director, and I will be discussing sample prep and batches. And it had been discussed a little bit, and I just wanted to go through some of the terminology. So when we talk about a batch, we're talking about a finished product manufactured according to regulations, GMP guidelines, or some people call it a standard processing unit. But a good test of a batch is that if it is divided, is nearly homogeneous or nearly the same, whether you divide it in half or divide it in quarters. So why do we want to sample? Why do we want good sampling protocols? To reduce bias in either a quantitative or qualitative analysis. In general, to follow USP 561 guidelines for botanics, if you have a container of a batch, you want to take from the upper, middle, and lower sections of each container. And when you're preparing samples by combining them, you don't want to alter them too much. You don't want to add or take away too much moisture. You don't want to significantly affect the sample. So the goal is to submit something that is a representative sample of what you have. As it was mentioned earlier, you know, if you just take the tippy topmost of the bud, you'll get a skewed result. So if you're trying to figure out the potency of a harvest and you go to this plant and you're like, I'm going to take this little leaf here and I'm going to dry it out, I'm going to send it, you're going to get like an astronomically high, you know, THC rating that doesn't represent what's actually in the plant. Here's a good diagram. These are trucks, but we can pretend that they're containers, and you can see these little dots represent samples. And the more you have, the more samples you need to take to verify the quality. There are ways of preparing samples. One way that people prepare samples is they take their batch and they cut it into quartering, and then they mix 2 of the quarters and submit that for testing. So they have a representative batch of what they're offering. And so to close, I just want to say that, you know, this quality improvement is is important for the industry. It's creating serious inroads into changing how management is done. Technology, innovations, I mean, really, it's really important for recognizing innovations in the industry. And organizations that hope to survive in this new industry, they better follow best practices, because that'll give them the capability to adjust to new standards. If you have nothing written down and you don't know how things are operating, you can't control the process. This is a really important thing. And I may disagree with one of our previous speakers is, you know, I don't really care, honestly, about the NIDA monopoly, but you know, they're not the only source of marijuana in this country, if you've noticed. But GLP, GMP, why are we doing this? Why do we care? We want standardized products to study them, right? I'll use Sioux Cicely. You know, we talked about this in Prague. Wouldn't it be great to have standardized sources of cannabis material for PTSD studies? This has already been done. I think, Michelle, you're talking about PROMIS later. So yeah, so this is already being understood. We want good laboratory, good manufacturing processes so that we can study the cannabis plant material. We can have a reliable source for researchers to use. Thank you very much for your time. Welcome the next speaker.
I'm Tony Young. I am General Counsel of the American Herbal Products Association. We are the trade association of the herbal products industry. I want to disabuse Michelle of my authorship of the petition to have the adverse event reporting by FDA. That was an APA project. Michael McGuffin and I worked on that, and the APA board approved it. And we essentially led the industry by saying the time has come for botanical or for dietary supplements to have an adverse event reporting system. No, I think I brought the wrong one. Yeah, and I don't have my pewter with me. That system, I believe, in 3 years, Congress passed a law adopting an adverse event reporting system. That system protects the dietary supplement industry from kind of press reports of adverse events that aren't documented in a system of filing. And I will get my proper slides so you'll all have access to them, I think, at the end of this, or yeah, probably by the end of this. And that system has protected us, I think, very well. And that's kind of what I'm going to talk to you about today is pharmacovigilance. But I'm going to start by addressing what I was asked to address, which is what lessons have we learned from the dietary supplement industry? And even before the dietary supplement law was passed, we had an incident with an ingredient called L-tryptophan, which is an amino acid and which was used by people basically to help them sleep. And it was used by a lot of consumers. And the company that manufactured it, the main supplier, was a company called Shodenko in Japan. And they decided that it would be appropriate, and this is something companies often do, to increase their yields by providing a purer product, so that instead of a 2% contaminant profile, you heard about contaminant profiles, it would have a 1% contaminant profile. And we never test contaminant profiles. People look at them, and I don't know that any testing would ever have turned up what happened. And what happened was that there's a disease called EMS, and only my slide would pronounce it properly because I'm not even going to try. But EMS is a debilitating or fatal disease. If you're debilitated, you get to live the rest of your life very, very challenged. If you die, you die. They had about 35 deaths and hundreds of people debilitated. $1.7 billion went from their company to the injured and to the lawyers. think about, and I always say this to the dietary supplement industry when I speak, think about suppliers of ingredients in your industry. How many of them have $1 million to stand behind their products if there's something wrong with them, even if they obviously don't know that it's wrong with them? So this is an example of where you tinker with a process of making an ingredient that has a track record of safety, and in tinkering, you seek to improve it, and something happens. Something bad happens. How many companies in this M and C industry are out there tinkering to create better products? The second big, I think, event for the supplement industry was ephedra, a very good herb, a botanical that created, through its chemistry, the drug ephedrine, the drug pseudoephedrine, and phenylpropanolamine, all drugs that are not so much on the market anymore, but they are very useful drugs over about 70 years. But the botanical was ephedra, and the botanical was very popular in the dietary supplement industry, beginning in about the mid-'80s, sold as a stimulant-type ingredient. And then after fen-phen, the diet drugs, went off the market, ephedra became a very popular diet ingredient in the supplement industry. And so you had an herb that had some limited use that all of a sudden became very, very widely used. And with that came adverse event reports starting in Texas, in a multi-level company that sold to a lot of people in Texas. Texas Department of Health got concerned. They started asking people about adverse effects, and they got back a lot of reports of, well, this happened, that happened. You know, very anecdotal, no kind of a study of any kind, but it piqued everyone's interest as to the safety of ephedra. FDA looked into it, and I testified at an FDA hearing. Mr. McGuffin did. Other trade associations did, and we tried to work out labeling and levels, ephedra levels, in products that FDA would then deem to be acceptable. Those discussions didn't go anywhere, in large part because a number of big marketing companies were moving into the area, and they didn't want to have limits, and without limits, the products took off very well in this diet industry, and more adverse events were reported to FDA. FDA then, after a young man died in Boston, on his couch, he was a student, he was an athlete, he was a person who worked out, pumped a lot of iron, and took a lot of ephedra products that they could trace because of those affinity cards that you get at your GNC or Whole Foods or whatever. Those affinity cards will track exactly what you're buying, and we assume you're using it all. And that started another investigation, and eventually in 2004 or 2005, ephedra was banned. The courts upheld that ban. And the lessons of ephedra are that people knowledgeable in the botanical industry understood this was a botanical that you needed to treat with respect. And yet the marketing people in the various companies were able essentially to co-opt the process for the usual reasons. Money talks and bullshit walks. And therefore, they co-opt the process. They continue to provide money and sell their products and make money. And so this good herb is no longer available to us. It's just no longer available in the U.S. All those companies, by the way, they went out of business. They went bankrupt. They're back in various forms now, but they all essentially went under with all the product liability litigation. And the product liability litigation is as a result of all those people who are injured or who are alleged they are injured and who hire lawyers. And once that legal process gets going, it wasn't 1.7 billion because there wasn't that much money there, and there wasn't anywhere near that kind of disastrous injury. But nonetheless, all of those companies went under. The rest of the industry paid extraordinarily high insurance rates for the next 2 or 3 years. So all of that success of those ephedra companies was paid for eventually by the entire industry. Sales were down generally in the industry. Don't lie to your government about adverse events. One of the companies took it upon itself to say, we have no adverse events. And then thousands and thousands of letters and telephone call intercepts, that kind of information that was all recorded in their files was disclosed. That guy went to jail for another reason, but he nonetheless went to jail. So that's an example of another one that took, oh, we got the right one? Whoa, you're the man. My consigliere, Michael McGuffin, was telling me what to do here, and I appreciate it. So this was a very long, expensive process that kept this kept the industry going for a while, and then brought it to an abrupt halt. Red yeast rice and lovastatin. When I tell this story, I say what I often say to the dietary supplement industry, but it's really true here. You all want to be in the drug business. That's what you're here for, right? Medical marijuana, that's drug. We got it, okay. Red yeast rice contains naturally occurring amounts of monocolin K, and when fermented to control that, you can bring it right up to the level of the Merck drug Mevacor with lovastatin. Or you can bring it to half the level. So you take twice as much to get that lovastatin. FDA held a rulemaking and found the red yeast rice products to be engineered, plainly described in their patents to control the monocolin K. I mean, all you would need to do was put all these patents together, and it was clear what this company intended to, did to, and they made a lot of money, and they kept it because there was never any recall or other kind of action, or even private lawsuits with regard to this product. It worked because it was just half the drug. I mean, you would expect it to work. So the FDA and the courts affirmed that it's a drug and you can't market it. Was it safe? Probably. But statin drugs are prescription for a reason. They seek to address cardiovascular disease, which you should be looking to a doctor for. We do that here. And there are side effects associated with statins. They're fairly well established, but you won't find them on the red yeast rice labels. But the lesson there is don't mess with Big Pharma. Don't try to emulate their drugs. They have lots of money, they have lots of lawyers, and they do not like competition. They do not like to be knocked off unless the people who are trying to knock them off do it through the drug procedures and pay all the high-ticket prices to go through there. Recent lessons in the supplement industry are engineered extracts of botanical components, various serious adverse event reports. These are mostly used in sports nutrition type products. Analysis of the data leads FDA to take regulatory action. And again, there are instances where people are trying to create drug-like compounds and arguing that they are truly botanicals. That dispute continues, but many of these products are off the market. So let's talk about CM product safety testing. Are you all doing animal testing? Well, that would be the little, medium size, and then the dogs, whatever. Are you doing human testing with protections for human subjects, where you disclose to the subjects all of the risks associated? Are you doing human testing in your own employees, friends, and families? That's often done. That's often done. It may violate the Treaty of Helsinki, which is an international treaty that governs how we test in humans, because humans have rights, and employees may be under a certain amount of job duress to do what you say? That's internationally recognized. Are you testing your products in your customers? Nobody takes them, or 1 or 2 people takes them, and then they're out there and you're testing in your customers. This is not uncommon, and I'm not suggesting it's only something might occur in the CM industry. It's just not uncommon. It doesn't occur in the drug industry. Pharmaceuticals are not tested in the customers. They're only tested in the customers insofar as 5,000 people in human tests is not necessarily predictive of what's going to happen after the first 1 million other people take the drug. But that's because they are very obviously powerful agents. This is a bad reason for pharmacovigilance, but it's a real reason for pharmacovigilance. Pharmacovigilance, it's the science and activities relating to the detection, assessment, evaluation, and prevention of adverse events or any other drug-related problem. And if you believe C&M products are drugs, if you do not believe, then you're not reading your own press releases, you're not reading the laws that are getting passed here. Medical marijuana, medical cannabinoid drugs, they're drugs. They're drugs. You're asking these legislatures to let you sell drugs. Treat them like drugs. Treat your patients, treat the customers with the respect. Now, I think you need pharmacovigilance nationally. I'm not talking about any state programs. That was a real horror story that I hear about the state of Washington with regard to laboratory studies, sampling plans. We're not going to have any sampling plans. Very basic item, but we're not going to have it. Maybe we'll have it now. But that's an example of why state requirements aren't going to address pharmacovigilance adequately. The big picture issue is to assure that there's a national repository of adverse event reports for evaluation. It's not a regulatory issue. You all are in this happy never land of not a lot of regulation. This is your business. This is your franchise. This is what you have devoted your careers to now. You're protecting your business. It's good science, it's good public policy and private policy, and it's respectful to the consumer and to the patients to keep track of everything and figure out if there's something happening out there, if there's something bumping in the night. Why it won't, all these products are different. Tablets, capsules, infusions, cookies, drinks, inhalation, water pipes, whatever is legal is in a particularly different. We could never sort this out. The formulas are proprietary. Pay no attention to the man behind the curtain. He has the secrets, she has the secrets. We won't tell you what's in it. And we won't tell you what we use to make it grow 2 or 3 times as fast. It's too expensive. Oh my goodness. You can't prove that my C or M product were the cause, the only cause, that they were, just because they were at the scene, just because there was a pipe in the car after the person was driving the wrong way on the freeway. That doesn't mean, and it was in their blood, that doesn't mean they got onto the freeway the wrong way because of some reaction to the product they were taking. Get over it. You want pharmacovigilance to work. You want to figure out the reasons to make it work. You want to discover problems and not repeat them. That's important to the preservation of this community. I like the story about the diethylene glycol because in 1938, the Food and Drug Act was passed when a very reputable drug company determined that the way to make sulfa drug, very newest, best, life-saving drugs available, to make it good, easy for children to take, would be to mix the sulfa drug with ethylene glycol. So it tastes good. Oh, that's good. About 100. About 100 lost their life. Okay? Big mistake. They probably could have tested it in 10 rats and figured out that people were going to die, but they didn't. They just said it tasted good. And it met all quality requirements because it was probably very good ethylene glycol. So you need to have an open exchange of information, a place where everyone can look and see what's going on, to see whether there are trends, things that are happening. It's necessary to assure product safety. Otherwise, it's just going to continue to be a mystery process. All the won't works are just challenges to be overcome by people smart enough to come as far as you've come. You're smart enough to overcome all those challenges. So you need to be your own FDA. You have to assure their safety. The states have no capacity to do it. There will be safety issues. There will not be standards created by the National Institute of Standards and Testing, because it's the National Institute, and you ain't national. The national government is watching to see how big a pothole you all are going to fall into, because the national government is not behind you. How many U.S. government representatives are there here at this meeting? They're not allowed to, officially. I asked a botanical scientist on yesterday if he was going to be at this meeting. He said, "Are you kidding? We can't go to meetings like this. We can't get funding to do that." They're not watching. They're only going to watch for the accidents. You have to be responsible for seeing that there are no accidents. What part of safe are we not understanding? Yeah, that's what we're talking about here. This is going to be a monumental effort. And being non-responsive, you'll then shift into the lawsuit area. So if I were selling these products at my store in Colorado or my dispensary in California, and if I were in the dietary supplement industry, I'd say, "Oh, by the way, how much product liability insurance do you carry? How much product liability insurance do people have? Is this even an insurable business? What's going to happen when some child or some elderly person or otherwise has a side effect with respect to one of these products and hires one of those thousands and thousands of lawyers who bring these kinds of lawsuits?" These things are going to happen. And the more successful you are, the more they're going to happen. And defending, if you have these things happening, and we found this with ephedra, if you have something happen in Washington, and I think I heard this afternoon, Washington, then Connecticut asked questions, and then some other state asked questions. And defending from one state to the other is something we did with ephedra, and it was a nightmare. Get on an airplane, go talk to some legislators who don't know the subject matter, and do your best to get them not to pass legislation. So let's all be careful out there. It's very important. I'm not going to be able to be at the APA Cannabis Committee meeting, but I want to suggest that those who attend think about gathering. There are cannabidiol drugs that are approved in the EU. The EU has first-class pharmacovigilance, absolutely mandatory for drug products. And what this industry ought to be doing is gathering the cannabidiol adverse event information and evaluating it and reporting, so that those of you who are developing those products can see what has happened in an organized system, a first-class system that has reported events, and so that you can see the kinds of things that you might see in the patients taking your products. Thank you very much.
Thanks so much to Tony Young, who will always amuse and horrify. Dr. Jehan Marcoux, Dr. Jeff Raber, and to Steph Scherer and Asa for having us on this panel.